Good afternoon everyone. Hello. How are you all? I hope each and every one of you is in the pink of health and happiness. So today we are going to learn about biomolecules. We'll first solve the questions. With the questions we are going to learn the concepts. Alright. So I am Ritya Koppalaramane. I will be teaching you biomolecules today. Alright. Everyone ready? So our first question is, which of the following is a nucleotide? We need to find out which is a nucleotide. Tell me which is a nucleotide here. So nucleotide ends with ialic acid or it ends with 8. It ends with 8. So it is uridine. INE ends, actually when INE ends, it is a nucleoside. It's a nucleoside. Nucleoside always ends with INE, ene, and nucleotide ends with ialic acid or 8. For example, adenylic acid, guanylic acid or guanylate, adenylate, that will be nucleotide. So here the option is B for Bombay, because uridine is a nucleoside, guanine, yes, guanosine is a nucleoside. So the answer is B. Which of the following is not a fatty acid? We need to find out which is not a fatty acid. Glutamic acid, it's a nucleotide, so it is not a fatty acid. Arachidonic acid, arachidonic acid is a fatty acid. Palmitic acid is a fatty acid. Lecithin, what is lecithin? It is a phospholipid, so it is not a fatty acid. Aspartic acid is a amino acid. Glutamic acid is also an amino acid, so this is also not the fatty acid. So it is, we need to find which is not, that is A, D and E are not the, A, D and E are not the fatty acid. That makes C as a right answer. Lecithin is a small molecular weight organic compound found in living tissues, is an example of, as I have already told you, lecithin is a phospholipid. Lecithin is a phospholipid. Given below are two statements. Statement 1, amino acid have a property of ionizable nature, yes, correct, and NH2 and COH groups hence have different structure at different pH. This is true. Amino acid can exist as Zwitter ion. Yes, they can exist as Zwitter ion, that is Zwitter ion form at acidic and basic pH. In the textbook, in the NCERT, they have given Zwitter ion in the neutral pH. But according to the concept, the Zwitter ion they can exist at the Zwitter ion form in the acidic as well as in the basic pH, which means the statement 2 is also correct, but according to NCERT they have given neutral pH, but if we can see the concept, it can be exist in the form of acid as well as in the basic pH, which makes statement 1 and 2 both are correct. Statement 1 and 2 both are correct. Both statement 1 and 2 are correct. Okay, and as it is given in the NCERT about the neutral, this statement can also be challenged with NTA if it comes. Read the following statement and choose the set of correct statements. Lecithin is found in the plasma membrane. Yes. Is a glycolipid? Is it a glycolipid? Actually, it's a phospholipid. It's a phospholipid. Saturated fatty acid possess one or more double bonds. Saturated actually forms single bond. It is already saturated, right? Single bond, which makes this as a wrong statement. Gingerly oil has lower melting point, hence it remains as oil in winter. Yes, as it has a lower melting point, even in the colder temperature, cool temperature, it can remain as oil in the, that is in the winter, that is cold temperature. This is correct. Lipids are generally insoluble in water. Yes, they are insoluble in water, but soluble in some of the organic solvents. Yes, this makes this statement correct. When the fatty acid is esterified with glycerol, monoglycerides are formed. Yeah, it can monoglycerides, diglycerides, triglycerides, anything can form. Yeah, it makes this statement also correct. We need to find set of correct statements here, which is C, D and E are the correct statements. C, D and E, D for Delhi is here, is a correct statement. I hope everybody is following. Any doubts? Any doubts till now? Yes. If there is no doubt, can I get a thumbs up from you all? Yes, thank you, Shamita. Following are the statements with reference to lipids. We need to find the correct statements here. We need to find the correct statements. Lipids having only single bonds are called unsaturated fatty acids. Lipids having the double bonds here. Unsaturated ones have double bonds. Double bonds. Double bonds. Lecithin is a phospholipid. Yes, we already know lecithin is a phospholipid. Trihydroxypropane is glycerol. Yes, trihydroxypropane is a glycerol. Palmitic acid has 20 carbon atoms. No, palmitic acid actually has 16 carbon atoms, which makes this statement wrong. Arachidonic acid has 16 carbon atoms. No, it has actually 20 carbon atoms. This also makes this statement wrong. So we got only B and C as the right answer. We need to find the correct answer here. So B and C only is the correct answer, which makes B for Bombay as a right option. Identify the basic amino acid from the following. So glutamic acid, in name itself there is an acid, which means this is not the basic one. Lysine, lysine is a, we need to find the basic amino acid. Arginine and lysine and arginine, yes, both are lysine and arginine. We know when we learn about molecular basis of inheritance, right? Lysine and arginine, actually it helps in coiling DNA. That is, DNA is negative. This is as acidic in nature, it will be wrapped around the basic amino acids, that is lysine. Yes, this is basic amino acid, is correct. We got the answer, that is B. Identify the statement which is incorrect. We need to find the incorrect statement. Glycine is an example of lipid. What is glycine? Glycine is a amino acid. It is not an example of lipid. Lecithin contains phosphorus atom in its structure. Yes, it is a phospholipid, so it has phosphorus in its structure. Okay, we got the wrong. Tyrosine possesses aromatic ring structure. Yes, it does correct the has the aromatic ring structure. Sulfur is an integral part of cysteine. Yes, sulfur is sulfur containing, cysteine is a amino acid which has, it's a sulfur containing amino acid, which makes D also right answer. We need to find the incorrect one. In the first statement itself we got the incorrect one, that is A is the incorrect statement. A typical fat molecule is made up of, typical fat molecule is made up of 3 glycerol molecules and 1 fatty acid molecule. No, 1 glycerol and 3 fatty acid, yes. So we know, right, what is actually fat? Fat is actually glycerol plus fatty acid. Fatty acid. What is glycerol? Glycerol is this one. It's a propane with, if we see, this is the glycerol. This is the glycerol. Now we esterify the fatty acid. What is fatty acid? Fatty acid is a long chain and at the end we have carboxyl group. Okay, if we esterified this fat with this, what will be, it will become the fat. How it will become? There will be okay, so there will be removal of this hydrogen, that is OH group from this, and the H from the that is, this was the COH, this was the fatty acid. From the OH, from the glycerol, the dehydration reaction takes place, that is minus H2, dehydration takes place, and this will bind to the carbon atom like this. So this is a typical, now there is no OH here, yes. So typical fat molecule, long chain of fatty acid. This one is a triglyceride. This is the triglyceride. If you see, there is one glycerol molecule, this is one glycerol molecule, this is one glycerol molecule, and we have 3 fatty acid molecules. This is one fatty acid, second fatty acid, third fatty acid. So this is the typical fat molecules, that is made up of one glycerol, this is glycerol, this is glycerol, and these are fatty acid. Together, these together make this triglyceride. This is a fat. This is the fat. I hope you understood. So this is B for Bombay is a right answer for this. A phosphoglyceride is always made up of. A phosphoglyceride is always made up of. It can be saturated and unsaturated fatty acid, yes, esterified with a phosphate group which is attached to glycerol molecule. No, it is not attached to the glycerol molecule. The only saturated fatty acid, no, it can be saturated, unsaturated, no. So the saturated and unsaturated is fine. Esterified to the glycerol molecule which has the phosphate group is attached, yes, it makes D for Delhi as a correct answer, phosphoglyceride. It can be, the fatty acid can be saturated as well as unsaturated, and it is esterified with the glycerol molecule which has phosphate group attached to it. So that makes the phosphoglyceride. Which of the following is not a secondary metabolite? Curcumin is a secondary metabolite. Yes. Morphine is the alkaloid which is secondary metabolite. Anthocyanin is a pigment, it is a secondary metabolite. What about the lecithin? Lecithin is a phospholipid. We find in the cell membrane. This is not the secondary metabolite. Which makes D for Delhi. See, curcumin, this is a secondary metabolite. Morphine is an alkaloid, is a secondary metabolite. Anthocyanin is a pigment, is a secondary metabolite. And lecithin is not the secondary metabolite, which makes D for Delhi as a correct answer. Match list 1 and 2. Choose the correct answer from the option. So again we need to, okay, these are the secondary metabolites here with, what are those? Okay, terpenoids, terpenes, diterpenes. So this is terpenoids, diterpenes are the terpenoids. Lectins, concavalin A is a lectin. Alkaloid, as I previously said, yes, codeine or morphine is a alkaloid, and toxin, ricin is a toxin, which makes A2B4 already there, C1, A2B4 C1, B4C1. Yes, D for Delhi as a correct answer. Now again match the following, and again there are set of secondary metabolites with others. Adenine is a purine. Anthocyanin is a pigment. is a secondary metabolite. Chitin fungal cell wall is made up of chitin. Chitin is a polysaccharide. Chitin is a polysaccharide. It is made up of N-acetyl glucosamine. N-acetyl glucosamine, that is NAG. This is NAG will be continuously forming the bond. NAG, like that. Okay. Chitin is made up of that. So it is a polysaccharide. Chitin is a polysaccharide. And codeine is a alkaloid. Morphine and codeine are both alkaloids. So which makes A4B1. That is B for Bombay as a right answer. Identify the incorrect pair. We need to identify the incorrect pair. Toxin, arvin. Yes, this is correct. Arvin is a toxin. Concavalin A is a lectin. We know it. Resin, actually resin is a toxin. Resin is a toxin, which makes C as a incorrect pair. And alkaloid, codeine is a alkaloid, which makes C is a correct. Here they have given ricin as a drug, but it's a toxin. Ricin is a toxin. Which of the following is not secondary metabolites? Okay. First, glucosamine is a amino acid and glucose. Actually, these are primary metabolites. Primary metabolites. Vinblastine, vincristine, curcumin is a drugs. Is a secondary metabolite. Rubber and gums, yes, secondary metabolite. Morphine and codeine, these are alkaloids. Alkaloids. Yes, this is a secondary metabolite. Now coming to what is not, they have asked not a secondary metabolite, which means amino acid and glucose are not the secondary metabolites. Concavalin A is a direct question. Which secondary metabolite is concavalin A? It's a lectin. Concavalin A is a lectin. Now we are going to match the following. Primary structure of the protein. Primary structure of, you know that, right? Primary structure of protein is just the polypeptide chain. This is polypeptide chain, where this is the amino acid, and it is the one amino acid is making the bond with another amino acid. This bond is called peptide bond. One amino acid making the bond, that is peptide bond, is established between one amino acid and its successive amino acid. This is primary structure. What is secondary structure? Even the same, the secondary structure of two types, that is alpha helix and beta pleated. Tertiary structure is the more common types for our enzyme structure, that is it is making a structure where it is bounded by sulfur bridges are there, and also hydrogen bonds are also formed. Hydrogen bonds are there, sulfur bridges are there. Okay, so it makes a pocket like structure. This is a pocket like structure where the substrate can come and bind. Most type of enzymes are of tertiary structure. So they make this thing. There will be a pocket like structure. Here there is a pocket like structure where a substrate, this can be a substrate can come and bind. This is substrate can come and bind. So most form, this is the enzyme. Now this protein, this whole thing is an enzyme. This is a substrate. So this pocket like structure here, we call it as active site. So the most enzymes are in tertiary structure. Quaternary structure means any two things, any like primary structure joining with a secondary structure, any of these forms, A, B, C, may any structure, tertiary structure with a secondary structure with a primary with a tertiary, anything. For example, tertiary structure, two tertiary structure are joined together, which makes a quaternary. Or maybe tertiary structure is joined with the secondary structure, maybe it beta pleated or alpha helix, that makes also secondary, or primary structure, that is polypeptide chain, it is joined with the secondary structure, that is beta pleated or maybe the alpha helix, anything which two of these forms, two of any of this ABC or three of anything which combine together to make the quaternary structure. Very good example of this is a hemoglobin. Hemoglobin, in this 2 alpha chain and 2 beta chain are joined together to make hemoglobin. So, very good example, that is hemoglobin. Hemoglobin is the example for quaternary structure. Now match it. Primary structure is just a polypeptide chain. Is just a polypeptide chain. Secondary structure is a alpha helix or beta pleated. Tertiary structure, yeah, disulfide bonds will be present or hydrogen bonds will be present in between. Quaternary structure, human hemoglobin. So, which makes A3B4, A3B4, that is A as a correct answer, C2D1, C2D1 as a correct answer. Now again match the following. GLUT4, here these are types of the proteins. Here they have given the proteins, you need to match it. What type of proteins are those? So, we know insulin is a hormone, we can match it. BK1 एक ही में C हो गया आंसर, लेकिन we'll match it again. Trypsin is an enzyme in digestion. There was previously chapter called digestion where we used to learn, okay, trypsin. Collagen is a intercellular ground structure. It's the most abundant animal protein. Okay, this is the most abundant. In animals, most abundant protein is collagen. You may ask, like, which is the most abundant protein in the whole, like, world में? That is Rubisco. Rubisco is the most abundant. When it comes to both plants and animals, wherever it is in the whole world, Rubisco is the most abundant. Even in plants also, everywhere. When it comes to animal, it's a collagen is the most abundant. Okay, so which makes D3. Then GLUT4. GLUT4 actually is the glucose transporter. It's a channel protein. Protein channel. Okay, where it transports glucose into the cell. So what is the answer to this? B1C2. Sorry, B1C. Okay, A4B1. We already got it, right? That is C as a correct answer. Now coming to the statement based questions. Statement 1. A protein is imagined as a line. The left end represented by the first amino acid C terminal. No, the left end is the N terminal. The left end is a N terminal, not the C terminal. So which makes this statement as a wrong statement. Adult hemoglobin consists of 4 subunits. 2 subunits are of A type, that is alpha subunit, 2 are alpha subunit, and the 2 subunits of B type, that is beta subunit. That is a quaternary structure. Quaternary structure we already know. That is a hemoglobin. Example is a hemoglobin which has 2 alpha chain and 2 beta chain. That's what the second statement says. Second statement is the right statement. So statement 1 is wrong, statement 2 is correct. Statement 1 is false, statement 2 is true, which makes A as a correct statement. So why the statement 1 is wrong? Because if you see, if you draw the protein structure, that is C carbon, that is alpha carbon, in which one is carboxyl group is attached, another one that is hydrogen atom, another one the R group is attached, and another one that is NH2, that is amine group is attached. So if you want to keep on adding the amino acids, then here we will be adding another this type of amino acids, that is NH2, C, H, here comes the R group and COOH. Now in between these two amino acids there will be the peptide bond formation. How the peptide bond will be formed? The peptide bond will be formed between these. Okay, there will be a dehydration reaction, that is minus H2O dehydration reaction, and here how I'll just write how it will be formed. C double bond O will be formed with this, and this is the peptide bond. With the N there is H will be formed here, this one. So this particular thing here, this one is the peptide bond. This is the peptide bond. If you see here, when another amino acid joins, it will be joined to here, then here, like that it goes on. So here if you see this amine group here, right, so this makes this strand as N-terminal. Always the left group, the left hand side here, the left end will be N-terminal, and here there is a carboxyl group. This is carboxyl group. Here it makes this as a C-terminal. That's why the statement 1 is wrong. Understood? The primary proteins are also called as polypeptide because they are also called as why they are called as polypeptides? Because they have the peptide bond formation. We already discussed this, peptide bond formation. So they can assume many conformation. No, primary doesn't have any conformations, and they are linear chain. That is true. But why are they called polypeptide? This statement doesn't explain why they are called polypeptides. They are polymers of peptide monomers. Yes, this statement is true, but it has to explain why it is called polypeptide. Peptide monomers are there, then why it is called polypeptide, it doesn't explain. The successive amino acids are joined by peptide bonds. Yes, this particular statement actually explains why it is called polypeptide. Why it is called polypeptide? This amino acids, one amino acid maybe, it's a monomer, yes, it's amino acid, another amino acids, another amino acid, another amino acid, another amino acid. So these are amino acids. These are joined with the help of something called peptide bond. That makes this as a polypeptide. So this is a monomer. One monomer is joined with another with the help of polypeptide. That's why it is called polypeptide chain. Now match the list 1 with the list 2, that is, okay, glycogen. You know what is glycogen? Glycogen is a stored product. Glycogen is a stored product in animals, Globulin is a antibody. Immunoglobulin, IgA, IgG, IgM, so these are antibodies, globulin. Steroids are hormones, you know it already, and thrombin is a biocatalyst. So which makes A as A4B3, C1D2. Yes, so A as a correct answer. Which one of the following is most abundant protein in animals? Now tell me, students, which one is this? Because we have already explained. This one. Discussed this. Tell me. Just unmute yourself and tell me. Let this be interactive class. अभी तक तो तुम लोगों ने कुछ भी नहीं बताया यार, क्या है ये? Yes, which one is the? Yes, Sakshi.
Collagen.
Yes, collagen. Collagen is the most abundant animal protein. Which of the glucose transporters is insulin dependent? Insulin in a previous slide when we were discussing about glucose transporters, there it was written GLUT4 as a glucose transporter. Transmembrane protein, when we were discussing about transmembrane protein, GLUT4. So this is a GLUT4 is a answer. GLUT4 is a transmembrane protein which where this that is insulin dependent for, okay, understood? Which of the following is not polymeric? Polymeric in a sense, the one individual monomeric unit is keep on joining, which makes a polymer. Okay, is nucleic acid a polymer? Because we have this sugar, phosphate, and this sugar is attached to the nitrogen base. This is one unit, and this again it is attached to phosphate, sugar, then sugar is to the nitrogen base, again to the phosphate, phosphodiester bond here, and again the sugar again nitrogen base with a glycosidic bond. So it makes a polymer, right? Yes, definitely the nucleic acid is a polymer. What about proteins? Polypeptide, anyway it is making the polymer proteins. Polysaccharides, glucose units. For example, if I take a starch, example starch. So it's the glucose units have joined together as a line with the help of 1,4 glycosidic linkage. So that also makes them as a polymeric. Now lipids, are lipids polymeric? No, they are not polymer. So which makes D4-DeLe as a non-polymeric, that is right answer. Which of the following is the least likely to be involved in stabilizing three-dimensional folding of most proteins? Which one? Least, we need to find which is the least likely. Okay, hydrophobic interaction, yes it is there. Hydrogen bonds, yes, for making the stabilization three-dimensional hydrogen bonds definitely required. Sulfur bridge is required. Electrostatic interaction, which means ions like positive and negative, yes, they definitely required. What about the ester bonds? Tell me, where the ester bonds can you find? You can find the ester bonds. Esterification of what? Esterification of fats. So you see these bonds in fats. Okay, so it has got nothing to do with here with the proteins. Okay, another one type of ester bonds you can see phosphodiester. Where do you find phosphodiester? Phosphodiester bond. Tell me students, where do you find phosphodiester bond?
Nucleic acids.
Nucleic acids, very good. Nucleic acid. So now this ester bond, does it have anything to do with the proteins? No, it doesn't have anything to do with the proteins. So which means B4 Bombay is a right answer. Now again match the following. Now we came to polysaccharides, nucleic and the types of bonds. What does the lipase do? Lipase, which degrade fats, right? So what lipase does? Actually it degrades the ester bond, fats, esters. Nuclease, phosphodiester is there. Protease, it degrades the, it lyses the, it lyses the peptide bond. Amylase, it lyses the glycosidic bond. So can you match it? A as 2, B as 4, A2, B4. A2, B4 is only in one, that is A. Correct? C1, D3. Yes, correct. A is the correct answer. Cellulose does not form blue color with iodine. So starch does. Starch actually forms. So you have done this experiment when you were small kids back in days. That the experiment is like, you take potato and you drop iodine on top of the potato, and suddenly potato turns blue black in color. So starch test, we call it a starch test, right? Or else if you grind the rice, you make a paste of the rice and you add iodine to it, it immediately turns blue black. What do you conclude? It has starch in it. Rice has starch in it. So why the starch gives blue black when iodine is added? Starch actually is a like this. Helical structure will be there with the starch. In this helical structure, the blue one, the iodine, it actually comes and traps here in the helical structure. When this does, actually this gives blue black in color. So one must have this helical structure, right? Like starch, it should have helical structure, then only you will get blue black. But when it comes to cellulose, it doesn't give any blue black. Why? Because it breaks down when iodine act with it? No. It is a disaccharide? No. Cellulose is a polysaccharide? No. It is a helical molecule. If it is a helical molecule, it should give blue black. So this is also not correct. It does not contain complex helix and hence cannot hold iodine molecule. So D4 Delhi will become the right answer to this. Any doubts regarding this? Tell me. Any doubts? No doubts? Yes, we will go to the next question. Again match the following. There are so many match the following. Protein. Which is a protein? Protein has which type of bond? Protein has polypeptide bond. Protein has polypeptide bond. Unsaturated fatty acid. Unsaturated fatty acid has double bonds. Nucleic acid, phosphodiester bond, and polysaccharide is a glycosidic bond. Easy question. So A4 B1. Yes, very good. Namdev, very good Namdev. Inulin. Inulin is a polymer of. Actually, this is inulin. Okay, it's a polysaccharide. It's a polysaccharide. What is it made up of? The polymer is made up of fructose. It's given in the NCERT. Okay, this is not insulin. This is inulin. Inulin is made up of the monomers of the joined monomers of fructose. It's a polymer. The fructose will be joined one after the other successively to make inulin. It's a very important concept here. Exoskeleton of arthropod is composed of. Now tell me what is the answer to this? Exoskeleton of arthropods are composed of?
Chitin.
Very good. Chitin. D4 Delhi is the correct answer. Match list 1 and 2. Protein. It's very easy. Would you match the list and tell me? Or is it the too bright is there? Are you able to see the screen, children?
Yes, ma'am.
Yes, right. Or should I decrease one second, hold on.
Option D.
Yes. Option D. Yes, very good. A is polypeptide. Unsaturated fatty acid. Oh, same thing. Again they have asked. Yes. So nucleic acid is a phosphodiester bond. Polysaccharide is a glycosidic bond. A4 B1. Yes, very good. Option D is the correct answer. Easy. Yes, it's easy. Okay, identify the substance which have glycosidic bond. Glycosidic bond. Where the glycosidic bonds? Polysaccharides. Polysaccharides or carbohydrates have the glycosidic bond and peptide bond. That is proteins. Proteins have polysaccharides and protein. Now glycerol. Glycerol is a. It's not. Trypsin is a protein. Okay, fine. That is not. Cellulose is a polysaccharide. Lecithin is a phospholipid. Inulin is a polysaccharide. Insulin is a protein. Chitin is a polysaccharide. Cholesterol is a lipid. Isn't it? So which makes C as a right answer. Understand? Because we need to find the substance which have glycosidic bond. Glycosidic bond in the sense they should be polysaccharides, and another one peptide bond they should be proteins. Here this is a lipid. Trypsin is a protein. So this makes A as wrong answer. Lecithin is a phospholipid, wrong. Inulin is a polysaccharide where fructose units are joined together, fructose. It is made up of polymer of fructose. Insulin is a protein. So which makes 3 as a right answer. Chitin is a polysaccharide, okay, but cholesterol is not a protein, it is a lipid. So it makes C as a right answer. Match the following. Aquaporins. Aquaporins are the protein channels. So if the water has to come in and out, the aquaporins actually help the water to come in and out of the cell. So this aquaporins are situated in the cell membrane, okay, cell membrane, transmembrane proteins. Aquaporins are transmembrane proteins. So which makes aquaporin as a protein which has polypeptide. Proteins are polypeptides. Asparagine, that is aspartic acid. Aspirin, that is a amino acid. Asparagine is a amino acid. Abscisic acid, it is a secondary metabolite, okay, secondary metabolite. It is a plant hormone or phytohormone. It is made up of, it is a derivative of carotenoids, we know that. Chitin is a polysaccharides. What it is made up of? It is made up of NAG, N-acetyl glucosamine. It is made up of NAG, which means this is one unit of NAG. NAG is again connected to another NAG, like that. It's a polymer like that. Okay, that is chitin. Chitin is a polysaccharides. So if we match 1, 3, 2, 1. Okay. Option D. 3, 4. Yes, very good, very good. Option D. D for Delhi as a correct answer. The two functional group characteristics of sugar are. What is the two functional group characteristics of sugar? Tell me. For example, if we write, we will write one simple sugar, that is glucose. 6. One is aldehyde group. CH2OH. So here balancing. So two functional group, one should be this one, carbonyl group, okay. The CHO we can write as C double bond O and H, right? It's the aldehyde group. And all other functional groups are, yes, very good, very good. Other functional groups are this hydroxyl group. Understood? So is there any phosphorus? We'll just eliminate it, okay. Option D is the correct answer as you already got it. Is there any methyl group here as a functional group? No. No, methyl again came. Then phosphate group? No. They have a carbonyl group, that is aldehyde group here as a functional group, and the hydroxyl group. Understood? So which makes D for Delhi as a right answer. Which one of the following statement is wrong? Tell me. Sucrose is a disaccharide. Yes, sucrose. What it is made up of? It is made up of 1 unit of glucose and 1 unit of fructose. So it's a disaccharide, yes. Cellulose is a polysaccharide. Yes. What is cellulose made up of? Actually, it is individual group glucose only, but it is beta glucose. Beta glucose. What is the type of bond here? It is beta 1, 4 glycosidic linkage. Glycosidic linkage. Okay, that is cellulose. When it comes to starch, I'll tell you just quick information, that's all. It is made up of alpha 1, 4 glycosidic linkage. Also we can see the starch is a branched, right? Starch is branched. So not only alpha 1, 4, wherever the branch is there alpha 1, 6 glycosidic linkage is there. alpha 1 comma 6 glycosidic linkage. This is alpha because starch is made up of alpha glucose, because it is made up of alpha glucose. Okay, uracil is a pyrimidine. Yes, it's a pyrimidine. Glycine is a sulfur containing amino acid. No, it's a very it's not a sulfur containing amino acid. What is glycine actually? Glycine is very simple structure it has. This is glycine, OH and NH2. This is glycine. This is alpha carbon and this is glycine. Does this have any sulfur compound? What are the sulfur containing amino acids? Actually, sulfur containing amino acids are cysteine and methionine. Cysteine and methionine are the sulfur containing amino acid. Glycine is a very simple amino acid, that here the R group is replaced by hydrogen, that is H hydrogen. This is a very simple amino acid, which makes D for Delhi as a right answer. The chitinous exoskeleton of arthropod is formed by the polymerization of, tell me. So, from this beginning of the lecture I have been telling you what chitin is made up of. Chitin is made up of NAG, right? What? Tell me which is the answer?
N-acetyl.
N-acetyl glucosamine, NAG, N-acetyl glucosamine, which makes it as a NAG. Okay, NAG, N-acetyl glucosamine. Easy. Which of the following biomolecules does not have phosphodiester bond? Which one of the biomolecules does not have phosphodiester bond? Tell me. Oh, sorry, does have. Does have. I'm really sorry. Okay, does have the phosphodiester bond. Polysaccharide. Huh? Phosphodiester. Does not need, does have.
Option C.
Option C.
Yeah, option C. Very good. So, does not have amino acid, does not have fatty acid, does not have. For fatty acids and glyceride, they have ester bonds, not phosphodiester. Okay, they have ester bonds here, esterification of fats happens here. So, here it is C, nucleic acid, they have phosphodiester bond. Understood? Pardon me students, I read the question wrong. Which one of the following is a non-reducing carbohydrate? Non-reducing carbohydrate.
Sucrose.
Yes, simple as that, isn't it? Yes. Sucrose is a non-reducing carbohydrate. It does not reduce in presence of Benedict's reagent. This is easy one. It's given in the textbook itself. Which one of the following graphs depicts the effect of substrate concentration on velocity of the enzyme catalyzed reaction? Tell me. Is the graph visible? Is the graph visible? Let me just. It's visible, right? Okay, shall I explain? Okay. So, if you see the graph here, okay, whatever. Okay, if you see the graph, if this is the velocity, I'm sorry. Velocity of the reaction, and this is substrate concentration. But why can I draw the graph here? Where is that? black. Okay, this is substrate concentration. Substrate concentration. मान के चलो. We have, for example, I have 10 substrates and 10 enzymes. I have 10 substrates and 10 enzymes. So if this is substrate 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 substrate constant. We have only 10 substrates, मान के चलो. And velocity of the reaction, this one is, so if one substrate bind to one enzyme, the velocity goes up. If two substrate bind to two enzymes, goes up. 3 substrate, 3 enzymes, like that it goes on, okay, until all my substrates and enzymes are totally bound, that is here, till here. After that, even if you add any more substrate, even if you add more substrate like this, if you keep on adding substrate here, keep on adding substrate, for example, again you add 10 substrate, you again add 10 substrate, what happens? Because the enzymes are already occupied, right? Even if you add more substrate, you have to add more enzyme to it, then only the graph goes up. Otherwise, if you have limited amount of enzymes and if you go on add the substrate, then that will be no use. It will attain the saturation here, this saturation. This saturation here, we call it as, this particular point here we call it as Vmax, that is Vmax, that is velocity is maximum here. That is a maximum velocity that enzyme substrate rate can attain, that the rate of the reaction can attain this maximum velocity. Maximum after that it cannot attain because we have only 10 enzymes. If we add more enzymes, then it can go up. With the more substrate, if we keep on adding, it can keep on going like this as a linear. But the enzymes are limited there. We have 10 substrate, it attained that one and it goes into the stationary phase. Okay, that is a Vmax. This particular thing here we call it as Vmax. And if we do the half of it, the Vmax, if we do the half of it, this is Vmax by 2, this particular point, that is Vmax by 2. If we draw a perpendicular to it, this particular point where it joins the substrate concentration, this particular point we call it as Km. Okay, this particular point we call it, this particular point we call it as Km. Okay, Michaelis constants, Michaelis constants, Michaelis constant. Okay, Km. So this particular point we call it as Km. Now tell me, if Km is low, Km decreases, what happens to the rate of reaction? Is it the good rate of reaction, which means the enzyme is very much, enzyme and substrate affinity is more or less? If the Km is low, if the Km is go low, I mean this way, if the Km is this side, if it goes on, then it can attain the velocity like this, right? It can attain the velocity like this. Now it can come to like this. So the graph would be like this, the graph would be like this, which means it is a good rate of reaction. Rate of reaction is good, like it is attaining the very steep. So if the Km decreases, enzyme is binding with the substrate, like good enzyme substrate binding, again good enzyme substrate binding. Okay, wait. Binding. So if the Km increases, this side, if the Km increases, if it goes this side, anyway, then which means the reaction is very slow. Can you see? The reaction is very slow. then only if you do the Vmax here, Vmax, V by 2, that Vmax by 2, then it will be like this. If the Km goes on increases, enzyme substrate binding is bad, isn't it? The enzyme is not feeling too good binding with the substrate, that's why it is very slow. Understood? This is the graph of the substrate concentration versus with velocity of the reaction. Any doubts regarding this, children? Tell me. Any doubts? You understood? Give a quick thumbs up if you understood this one. Thank you, Sakshi. Yes, yes. So now just answer this question after this. Answer the question. Enzyme's concentration on velocity of the enzyme catalyzed reaction. Is this correct? It goes on like this? No, right? This is wrong. The Vmax, is it how the Vmax is attained? No. Is it how the Vmax is attained? No. This is how the Vmax is attained, right? Just now we discussed in this particular thing, we discussed the same thing, isn't it? Did you not understood or not answering the question? Why? It's such an easy question. Directly the graph from the NCERT book is given here. Okay. The enzyme that catalyzes the removal of groups from the substrate by mechanism other than hydrolysis, leaving double bonds, are known as, actually, if it is like this, C is with X and C with Y. This statement is directly from the NCERT textbook. This statement is directly from the NCERT textbook. Okay, which type of substrate you, which type of enzyme you add so that we get the double bond plus X plus Y or XY, anything is fine, XY. So, what it is? It is actually lyases. One of the example of lyases, aldolase. Example is aldolase. So, this is a direct NCERT textbook statement. Regarding the catalytic cycle of an enzyme action, select the correct sequential steps. We need to select. So, here before that I will just explain it to you how. This is the enzyme, for example, this is the enzyme. Okay, enzyme is this one. Okay, enzyme, and we have substrate here plus substrate. Okay, this is the substrate and this is the enzyme. So, what it will be? Now, the enzyme substrate reaction takes place. Enzyme substrate complex. Enzyme substrate complex. This is E plus S give rise to enzyme substrate complex. Now, the enzyme will turn the substrate, this is enzyme, this is substrate, turn the substrate into product, isn't it? The enzyme will be intact like this. Okay, this is the enzyme. Now, this substrate will be converted to product. This one. Okay, for example, this is a product. It has converted into product. It's no more like a substrate. It's a different one. Okay, we can make it even more different. Yes. So now this is the enzyme plus this is product. Got it? So this is the enzyme substrate reaction, or else this you can call it as lock and key mechanism. Lock and key mechanism. Lock and key mechanism. isn't it? So for a particular lock you need a particular key. In the same way, for a particular substrate you need particular enzyme, or particular enzyme you need particular substrate. It's not like any substrate can react with any enzyme, or any enzyme can convert any substrate into product. It is not like that. It's a particular things, that is enzyme substrate reaction. Now based on this, tell me, substrate enzyme complex, okay, free enzyme ready to bind, it is the last step, free enzyme. Now this free enzyme, this is ready to bind, this is ready to bind to the next substrate. This is the last step now. Release of products, no, chemical bonds of the substrate broken, substrate bind to active site. First step is, this is the first step, the substrate binding to the active site, this is the first step. Okay, chemical bonds of the substrate are broken. Here the bond formation will be there, the chemical bonds will be broken. This will be the second step, that is enzyme substrate complex. After that, enzyme, okay, wait. Now here the enzyme substrate complex formation are taken place. This statement is there. I am really sorry. Now enzyme substrate complex has taken place. After that, chemical bonds of the substrate are broken. This will be the third one. And the free, okay, release of product, this will be the fourth, and free enzyme, this will be the fifth. So now CAD, okay, EADCB, yes, C is the correct answer. Any doubts? Any doubts? Yes. So we are going for the next question. Inhibition of succinic dehydrogenase enzyme by malonate is a classical example of, this is a classical example of competitive inhibition. This is a classical example of competitive inhibition. This malonate actually resembles this one, okay. Resembles succinate. For that's why this malonate will go and bind with the succinate dehydrogenase. It will go and bind with the succinate dehydrogenase. This succinate dehydrogen, as it resembles the actual substrate, now it cannot make the differentiation between this malonate and the original, that succinate. That's why it will be the competitive inhibition. If you increase the succinate, then in the competitive inhibition, if you increase the actual substrate, then the reaction can go in a very good pace. So now this is the classical example of competitive inhibition. And this malonate, this is a inhibitor. This is a inhibitor. The cofactor of an enzyme carboxypeptidases. Tell me, which is the cofactor of the carboxypeptidase enzyme? It is given in the NCERT. Directly line it is given. The last topic. Tell me. Tell me, children.
Option B.
No, niacin. No, actually, niacin is a coenzyme.
No, ma'am. Option B.
B, heme. Carboxypeptidase. Heme is the actually prosthetic group for which one? It's a prosthetic group. Heme is a prosthetic group. For peroxidase, catalase, enzymes. Okay, these are enzymes. Actually, zinc is a metal ion, which will be with the carboxypeptidase as a cofactor. Okay, this one is zinc. Any doubts? This statement directly from the NCERT. Okay, children. Read the NCERT line by line. Malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of succinic dehydrogenase. Very good. The given below are two statements. Low temperature preserves the enzyme in temporary inactive stage. Yes, it's make that at the low temperature the enzymes will be inactive. This is correct. State where the temperature destroys the enzymatic activity because proteins are denatured. At the high heat, the proteins will be denatured. We cannot retrieve it back. So this is the correct answer. I mean correct statement. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of enzyme, it is known as competitive inhibition or competitive inhibitor. Subhash? 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 The inhibitor of the catalytic activity is malonate. Possess peptide bond is a collagen. Cell wall material of the fungi is chitin, and secondary metabolite is a ricin. So now just match it. 1 A2 B4 2 4. That is D4 Delhi as a correct answer. The organic compound that is tightly bound to enzyme protein is the prosthetic group. Is it correct? It's the prosthetic group. Actually, prosthetic group is an organic compound and it is tightly bound to the enzyme. Yes, this is correct. This particular statement is correct. The complete catalytic active enzyme with its bound prosthetic group is called apoenzyme. No, actually, the complete catalytic active enzyme is a hollow enzyme. Hollow enzyme. It is made up of apoenzyme plus cofactor. Apoenzyme plus cofactor. What is this apoenzyme? Actually, it is the protein moiety. What is this cofactor? This cofactor can be a coenzyme, prosthetic group, or it can be a metal ion. For example of coenzymes here, I have already given you the examples, that is again we will discuss. The coenzyme can be the vitamins that can be NAD or NADP, okay. And prosthetic group, the prosthetic group can be heme. Metal ion can be the zinc, which we have already discussed. Now, is it called apoenzyme? The complete catalytic enzyme here, this complete catalytic enzyme we call it as hollow enzyme, which bound with the prosthetic group. Actually, apoenzyme binding with the prosthetic group called hollow enzyme. So this makes this as a wrong. So A is true, B is false. B for Bombay is the correct answer. Understood, students? Understood? Any doubts? Just tell me any doubts. Which one of the following statement is correct with reference to the enzymes? Oh, just we discussed. Just tell me the answer. Just discussed. Tell me the answer.
Option B.
Option B. Hollow enzyme is equal to apoenzyme plus cofactor. Okay, apoenzyme. Apoenzyme means it is made up of protein. Okay, protein. And what are the cofactors? Cofactors can be coenzymes, metal ions, metal ions or prosthetic groups. So yes, B for Bombay is the right answer. Easy. Now, which one of the following describes the given graph? Okay, there are two types of reaction, that is endothermic reaction, exothermic reaction. Okay, I will draw the graph here for the exothermic reaction. Okay, if this is the substrate and if this is the product, when it is becoming the substrate to the product, it has lost its energy. Okay, this is exothermic. Okay, and from the path, when it's become from the substrate to the product, when it is becoming in middle, it has gained the energy gained is a endothermic. This has lost the energy, which is exothermic, which has gained the energy is a endothermic. See, if this is the energy, the substrate in this energy level, the substrate is in this energy level, the product is in this energy level, this is an endothermic reaction. The substrate is in this energy level, it has lost the energy becoming a product, this is endothermic. Now what is this? Now this is the substrate in this energy level and the product is in this energy level, so which means it has lost the energy during the process, which makes this as a exothermic reaction. And also we need to find the, okay, this is A, this is with the enzyme. The B here, this is without enzyme. Tell me the answer. Endothermic to mark it wrong. Exothermic reaction with energy A in the absence of enzyme. A actually is in the presence of enzymes, which means this is also wrong. Option D. Very good. Exothermic reaction, we know this is the exothermic reaction with the energy A in the presence of enzyme and B in the absence of enzyme. Got it? Very good. The non-proteinaceous enzyme is, we say most of the enzymes are proteins, but some are nucleic acid. Which is that? Tell me. We have repeatedly discussed this. Tell me. The non-proteinaceous enzyme, there is something called ribozyme. In the previous class also I told you. What is it? Actually it is a RNA. It's a RNA which is behaving as a enzyme. Yes, ribozyme. Very good. So this makes D as a correct answer. Which of the following statement is incorrect? We need to find the statement which is incorrect. The competitive inhibitor does not affect the rate of breakdown. Yes, it does not affect the rate of breakdown of the enzyme substrate complex. The presence of competitive inhibitor decrease the Km value? No, it doesn't. The competitive inhibitor reacts with the enzyme to form enzyme inhibitor complex. Is in the enzyme inhibitor complex. We need to find which is incorrect. Okay. The enzyme inhibition, the inhibitor molecule is not chemically changed. The inhibitor molecule by the enzyme. So this is also called, it is not chemically changed. It is just going and binding. Okay, we will go back. The enzyme inhibitor does not affect the rate of the breakdown of enzyme substrate complex. The presence of competitive inhibitor decreases the K. Okay, it doesn't decreases. It is given decreases. So it doesn't decreases. Doesn't decrease the enzyme substrate complex, which makes this as a incorrect one. Okay. Actually it doesn't decreases the Km value of any enzyme. Understood? The enzyme, the competitive inhibitor. And competitive inhibitor, that inhibitor does not get chemically changed. This is also correct. And competitive inhibitor act as an enzyme to form an enzyme inhibitor complex, which means if this is the enzyme, if any this thing is coming, inhibitor, for example this is the inhibitor, samjho, okay, this is the inhibitor, it just goes and binds with this, okay, that makes this enzyme inhibitor complex, it goes and binds enzyme inhibitor complex, but will it change to product? No, it doesn't change to product. Understood? So which means A, C and D are correct, but B is a wrong answer. we need to find incorrect one, that is B for Bombay is a incorrect option. Incorrect statement, so that is the correct option. Select the option which is not correct with respect to enzyme action, which is not correct. We need to find again incorrect one, not correct. Malonate is a competitive inhibitor of succinic dehydrogenase. Yes, this is correct. Substrate binds with enzyme in its active site. Yes, it does bind with the active site. Addition of lot of succinate does not reverse the inhibition of succinic dehydrogenase by malonate. Now, competitive inhibition, competitive, which means this malonate is giving the competition to the succinate for the binding site of succinate dehydrogenase. If you increase the succinate very much, for example, you increase the concentration of succinate very much, then the succinate will definitely bind to the succinate dehydrogenase, not the malonate. Okay. So, here they have given does not reverse, actually it reverses. So, this is the wrong one. We will go through D also. The non-competitive inhibitor binds the enzyme at the site distinct from the active site. Yes. In non-competitive inhibition में क्या होता है? If this is the enzyme, I will tell you, if this is the enzyme and if this is the active site, if this is the active site, this will be the non-competitive inhibitor will come and bind to this particular site. This is non-competitive inhibitor. When this non-competitive inhibitor, this is inhibitor comes and bind to the enzyme in the different, now it will change the active site. When it changes the active site, the real enzyme, the substrate which has to bind, if this is a substrate, it has to come and bind here. Now it cannot bind here. This is non-competitive inhibition. This is anyway inhibitor, but it is not binding here. But in competitive inhibitor, the malonate is coming and binding here with where the substrate has to bind. Understood? So this is also correct. D is also correct. But about C is the wrong answer. Understood? The essential chemical components of many coenzymes are, you know it already, tell me, it is, example, NAD, it is a vitamin, coenzyme, this is coenzyme, this is the coenzyme. The transition state structure of the substrate formed during the enzyme reaction is, actually it is transient and unstable. It cannot be permanent. See, enzyme, I said no, enzyme substrate complex, this, if this is binding here, the enzyme substrate complex, I will draw the complex here, wait. If this is the enzyme substrate complex, this is substrate, this is enzyme, it cannot be permanent. If that is permanent, then will the product formation will be taking place? No, it should not be permanent. And if it is stable also, will the product formation taking place? The bond will be stable, it has to break the bond, it should be unstable. So D for Delhi is a correct answer. Understood? So yes, this is the last question. We have solved around 13 years of the previous year question. I hope everybody have got the confidence with regarding to biomolecule chapter. Is it like, did you got the confidence? Just thumbs up. Thank you, Shamita. Thank you, Pushpa. Yes, thank you so much, everybody. So this is the end of the chapter of biomolecules. We have an announcement. Tomorrow there is a mock test. Please come prepared. So that is the end of the session for today. Thank you one and all.